All right, everybody. This is Harper Van Steenhouse again. I'll be hosting the webinar here on behalf of BioNano Genomics. Thank you, everybody, for attending. We have an excellent presentation today by Mike Rossi from Emory University. And he is going to be talking about some work that he's done in collaboration with us looking at multiple myeloma and using BioNano Genomics Irys technology to do so. Before I hand it over to Mike, however, I'd like to give everybody a little bit of introduction to the technology that Mike is going to be talking about later on. So without further ado, I'll get into a couple of slides. During the talk, feel free to submit questions. You can do that through the Q&A box. You just type in your question, and that goes directly to the panelists, including myself. During my section, I can answer questions at the end, and we can provide time at the end of Mike's presentation also to give some answers to questions as they come in. So appreciate all the questions you have. Again, my name is Harper Van Steenhouse, and the technology and product that we're going to be talking today is the Irys platform. So what you're seeing in this movie right here is an individual molecule moving through the nanochannels on the Irys chip. This is really the heart and soul of the Irys platform. And it's the way that we are able to provide all of you with a new platform, a new tool that allows you to have a more comprehensive understanding of genome biology and a better visualization of the genome on the basis of extremely long individual molecule analysis of DNA. So with that, you're able to generate higher quality, more complete finished de novo genomes in faster time, as well as use this information to get more meaningful biology based on structural variation, the ability to see repeats and architecture in the genome that's not possible with any other technology. The way that Irys does this is giving you a different view, and we think it's a better view of the genome. And it's on a different scale than you get with sequencing. So in sequencing, you get base pair resolution, but you're unable to see things on the scale of kilobases and megabases because of the repetitive and complex nature of genomes. And so we measure directly those sorts of variations. We do this by retaining long-range contiguity, even in repeats and structural variants, because we don't fragment DNA up front. And as you'll see, the technology in the chip allows for extremely uniform linearization and stretching of this single molecule DNA so that you can get very highly precise and reproducible measurements. We incorporate a labeling scheme that allows sufficient uniqueness to assemble de novo complex, large genomes, as well as identify variation in a highly precise manner. And then, as you'll see, just as a practical manner, multi-cycle imaging and repeated analysis of a single sample automatically through the instrument allows you to get massively parallel throughput that's really required for big genome analysis. The advantage of keeping molecules intact right from the beginning all the way through data analysis is that we're able to generate more complete genomes and more contiguous information on the back end. This is just a quick demonstration of a moderate-sized genome. It's a 12 megabase streptomyces genome that a collaborator worked on. And you can see that with short read technology, you're able to assemble a very fragmented genome, hundreds of contigs, and relatively incomplete at a whole genome scale. And that's the first row. The second row shows you what would happen if you added longer range information in the form of cosmids, and you improve the assembly somewhat, increasing the coverage and reducing the contigs down to about 100. So-called long-reach third-generation sequencing does even better, and here you can get a decent assembly, but you still have about 20 contigs. Whereas with BioNano, when you're starting with megabase-sized DNA and you're reading individual molecules that are hundreds of kilobases long, you're able to assemble a single contig covering the whole genome. and keep all of the genome contiguous. When you have that sort of scale and contiguity, you can really take a broad look at the genome and start asking serious questions about biology that you don't have access to with other technologies. And that is looking at structural variation and rearrangements and translocations that Mike Rossi is going to be talking about in a couple minutes. And in addition, for scaffolding and validation of assemblies and really adding information to de novo genomes that you have no other way of getting, which is really identifying gaps in a comprehensive whole genome manner, and then diving down into haplotype differences between, because you have access to the single molecule reads. So we've done this, we've used this technology, and our customers have used this technology on a variety of different organisms. Certainly today, at the center of Mike's talk is the human. But this technology works just fine for genomes of all scales, from bacteria all the way up to crop plants, a variety of pests, fungi, mammal-sized genomes, as well as a variety of model organisms that, while have very good reference genomes, we can still contribute quite a bit of information, even though sequence information has been used for years in some of these model organisms. So with that intro, I'll just talk a few minutes about how the technology works that I've alluded to. This is what the platform is. This is the Irys platform. On the left, you'll see the Irys Prep reagent kit. This is a kit that is used for extraction of extremely long DNA and the subsequent labeling of it in a sequence-specific manner. Then the sample is loaded onto the Irys chip. The Irys chip goes into the Irys instrument. The Irys instrument is a high-resolution automated scanning microscope that collects image data as well as electrophoresis the DNA into the channel. I'll show you a close-up of what that looks like in a subsequent slide. And then we also offer a host of comprehensive software, Irys View, for collection of the data, organizing, as well as analysis. As I mentioned in the first movie that I showed, confining single molecules, isolating them, and getting them into nanochannels is really the key of what we do. And this is how we're able to offer this view that I've been promising you. The key depicted in this slide and a subsequent movie is that while DNA in solution is coiled up in simply a mess, as a charged long polymer, we need to be able to get individual molecules isolated as well as linearized so that we can make direct measurements across long spans in the genome. And we do that by electrophoresing the DNA through a variety of micro and nanostructures and ultimately into a nanochannel where they're isolated. Once in a nanochannel, molecules are linearized, they can't fold over, and they can't overlap each other. And I'll show you what this looks like in an image. The Irys Chip V2 is our current version of the product. This was released at ASHG just about a month ago. And this is our high-throughput human-capable chip. It takes 3 chips to analyze a human to 30x depth, and we have 2 different flow cells on each Irys chip that can be used independently if you have a smaller size genome. Inside each of these chips are 26,000 nanochannels, and you'll see what this looks like in a movie coming up. So the workflow is fairly straightforward. You extract long DNA, and by long I mean hundreds of kilobases to megabase sized DNA, from cells or dissociated tissue. And the common path here is to use a gel plug method to keep the DNA in a matrix so that it does not get fragmented. So once you have this unfragmented long DNA isolated, it goes into the labeling reaction. There's lots of different ways you can label DNA to put it through the system. Our first kit uses this nicking and labeling approach that's described in this slide. Here, we take long DNA, use a nicking endonuclease, and simply similar to a restriction enzyme, but it cuts only one strand. So it creates a single strand nick at a specific 7-base motif throughout the genome. We use a polymerase to incorporate a green fluorescently tagged nucleotide at each of these 7 base motifs throughout the genome, and then you take that labeled DNA and put it into the chip. The picture on the right shows you what this looks like. You've got blue molecules with green labels superimposed on top of them. It's this unique signature that will be used later on by the informatics in the system to uniquely assemble de novo your genome from this pattern and use it to add context, whether it be comparison to a reference or comparison between individuals for structural variation. So that was the end game there, was the picture of individual molecules in channels. This is how this happens, is depicted in this movie. You pipette the sample into the chip, and then the instrument takes over and electrophoretically moves the DNA. In this picture, in this movie, it's going to be from left to right. You see the DNA labeled in green on the left in solution. It's all glommed up together. Really not separate molecules at all. You can't tell what's going on. But as you'll see as the movie progresses, the DNA will move into the chip and move through a variety of different structures from large to small to isolate and unwind the DNA and ultimately get them into, get them into individual channels so you can image single molecules. So now they're moving into microchannels where they're becoming isolated from each other. They'll pause at an interface and then move into the nanochannels where now you're imaging single molecules of DNA separate from each other in independent channels, perfectly linearized and stretched at the same 85% of theoretical maximum so that a pixel width is the same number of base pairs every time. I'll let this play through one more time. So this is a looping movie, but on the instrument, what would happen is once the molecules make it into the channels, the current turns off and the imaging begins, collecting images of the entire array of those 13,000 nanochannels per flow cell and collecting that data, and then it flows DNA in again to do another round of cycling and imaging. And that's shown in this animation here. Each image set is collected by the system, handed off to the analysis module that turns these into digital depictions of molecules, essentially these green dots on the blue strings, and then the assembly algorithm takes those and performs essentially an overlap layout consensus assembly to do de novo genome map building. So you end up with a consensus map now that you can use for a variety of applications. As I mentioned, depending on what you compare your genome map against, whether it be a reference or another individual, you can do different types of structural variation detection. If you compare your sequence contigs that are essentially digested in silico by our software, you can do scaffolding and anchoring of your assembly. Structural variation simply looks depicted in this slide as differences between two different maps, either the addition of labels in the case of insertion or the spreading apart of labels in a smaller insertion, or in the case of a deletion, you have sites that disappear or get closer together. So we're able to detect a wide range of sizes from very small to very large, and we don't use any reference in our de novo assembly, so you don't have a bias as you would in arrays or resequencing approaches. Further, insertions and deletions are detected in a symmetrical and opposite way, and so you don't end up with a bias of being able to detect deletions better than insertions in this method. I mentioned the ability to see haplotypes by virtue of the individual molecules. I've been showing you kind of the map level over the last couple of slides. Now we're diving in from each of these maps, the read pileups that comprise those maps to make the consensus. And you can see here at this level, while the outside of the blue box, the genome is identical, inside the blue box there's several differences that separate these two haplotypes. And so we build these de novo maps separately for each haplotype and identify those, along with the single molecule support for that. Okay, so that's the end of the technology. I'm going to hand it over to Mike now for him to talk about how he's been using this technology. But I'll just sum up with a few things that I showed. Really, the ability to get this individual molecule data at an extremely long range allows you to visualize and understand genomes more completely than you can with fragmented and relatively indirect methods to get the additional genomic architecture and organization information that you can't get any other way to do structural variation and de novo assemblies. So in addition to asking any questions during this, feel free to email us at any time and follow us on Twitter to stay in touch with us. We're happy to answer any questions you may have at any time. So just wait 1 second. We'll hand over a presentation to Mike Rossi. Are you there, Mike? Are you muted, Mike? We don't hear you.
Sorry. Yeah, I was muted. Sorry.
Okay,
so what I'm going to talk about today is a pilot project that we've done in a disease site that's important at my institution and many other institutions, multiple myeloma. The reason why I put multiple myeloma first is because I think this is an ideal fit with this technology. I think other hematological malignancies are also a good fit with this technology, but for us, I'll explain why this is important and why we really want to see this technology advance. So for those of you in the audience who aren't familiar with multiple myeloma, it's a plasma B-cell malignancy. The current way to evaluate this disease and disease progression is with conventional cytogenetics, G-band karyotype, and FISH. And it's really these traditional cytogenetic technologies that are used to assess risk and disease progression in these patients. Most importantly, there's some translocation events, 414, 1416, and copy number abnormalities like deletion of 17 and 13 that are associated with a clinical course with these patients and very well characterized in the literature. And so at our institution, we have a very strong clinical unit, and we process about 1,000 multiple myeloma samples per year at the routine G-band and FISH karyotype level. And what we're really in the process of trying to do is, and like many other institutions, is trying to figure out exactly how we merge old technology with new technology and where the combination of the two meet. So we continue to do karyotype and FISH as a clinical test, but on the research side we do RNA-seq and copy number and a number of next-gen sequencing methodologies to really try to get at it, what's driving the disease in individual patients. And so we've embarked on testing a number of patients that are fully consented as part of clinical trials for us to do comprehensive genomic analysis, and what this slide shows is the first patient we had. This was recently presented at ASH, which shows a pretty typical clinical course of a patient who is diagnosed, undergoes disease progression several years after diagnosis, undergoes a stem cell transplant, eventually fails therapy, gets put on clinical trials, and goes through the succession of clinical trials. For this particular patient, you can see at the top of the figure right here, hopefully you can see my arrow, which says there's an arrow here that says RNA-seq, SNP copy number, and aplastic cancer. So back in 2012, last year, October of last year, this patient came in as they were entering their second clinical trial, and we were able to obtain a bone marrow isolate for specific research testing. And we took that isolate, we used enrichment of CD138 for plasma B cells. We extracted RNA and DNA, and then we did RNA-seq, SNP copy number, and targeted mutation panel on that patient. The patient then went into trial, and we obtained some very interesting data, both in terms of multiple copy number abnormalities. Down here you can actually see where the copy number variation is. Up here you can see where our FISH probes are, so we have a very large FISH panel that we've transitioned to running copy number arrays with, just because we can't see all the copy number abnormalities that are happening in these patients without doing something more comprehensive. We also run a mutation panel. Initially we ran the an ion torrent panel, and now we run a Illumina MySeq panel. And this is very important because we're able to detect clonal populations. This particular patient had a mixed clonal population containing both NRAS and KRAS mutants, and there's quite a bit of information at ASH this year about a number of patients that have been characterized that have KRAS and NRAS or BRAF and KRAS mutations that appear to exist as different clonal populations. So using a combination of DNA mutation panels, deep sequencing panels, plus SNP copy number arrays, we're really able to look at clonality. And then we use RNA-seq really to be able to get at differential gene expression and fusions. This copy number mutation panel cannot give us fusions or differential gene expression, of course, and so RNA-seq is really a tool of interest that we rely very heavily on. We think it's a very powerful tool. Can also give us coding mutants, which we think is very reliable. The only problem with that approach is that we find lots of aberrations that happen, translocations, potentially interstitial rearrangements that happen that create these fusions. And so this is a circus plot showing potential translocation between chromosome 1 and 10, potential translocation between chromosome 3 and 13, and then several inversions that or fusions that may happen within a specific chromosome arm. And we're usually stuck to try to figure out how to confirm all these changes. And so you can see here a table of all the potential fusions that we detected in this one patient. A number of these we have to go back and try to confirm by PCR, and usually this is just the simple endpoint PCR, but it's the matter of going back, redesigning primers. Depending on the complexity of the rearrangement, you may have to design several primer sets to be able to know exactly what the fusion product is and to test it appropriately. And you can see that there are a number of false positives and a number of changes that happen that are from pseudogene to pseudogene, so they're the wrong call at the sequencing level. So it's just there's homology between one gene and another partner, and so they're difficult to characterize. And we use copy number to try to tie this together, and you could use exome sequencing as well. We find copy number is a little as lower cost and a little bit more useful. And so you can see here this is the patient's chromosome 1, and you can see there's a large deletion here, and then there are all these kind of small spikes that happen that indicate potential for copy number abnormalities. You can see that there's this one little line right here where the arrow points to. There are these two genes, and this segment is a little over 100 kilobases, and we actually were able to detect a fusion between this gene and this gene. So the breakpoint actually happened 5' to 3', including this gene, and it includes what appears to be a copy number abnormality. And some of these are very well characterized. We had another one very similar to this on chromosome this was within one chromosome arm, and this shows a fusion. And sometimes you have these copy number abnormalities that actually create this kind of false fusion that's actually a benign change. And then you have these other changes where it's within another chromosome, so this is chromosome 20, where you have these copy number abnormalities, and it's really hard to tell is this caused by a somatic change, or is this caused by some type of benign constitutional abnormality. And the context and the context of what's happening in these changes is very hard to interpret. And so it often requires getting in normal DNA, going through multiple changes. So we're really trying to look for technologies that kind of summarize everything, and I think that that's really where BioNano Genomics comes in. And so we approached BioNano Genomics with this proof of concept experiment where we took a very well characterized multiple myeloma cell line, KMS11, and we allowed them to run their technology to fully characterize what was happening. And as Harper went over, the resolution of this technology is really to be able to see structural rearrangements at a larger contig level. So rather than the 100 to 200 base pair resolution that we're looking at with next generation sequencing, really being able to see sequence context with these changes. And so as part of a internal grant that was sponsored by our Cancer Institute, we were developing our next generation sequencing pipelines, and what we really wanted to do was kind of characterize what was happening with microarrays and accommodate that transition into next generation sequencing. So we took three very well characterized multiple myeloma cell lines, RPMI 8226, KMS11, and MM1S, and we ran the full gambit of microRNA technologies, copy number, gene expression, methylation, and microRNAs. And we complemented that with all the next generation technologies, RNA-seq, paired end, and mate paired whole genome sequencing, as well as some microRNA sequencing that we did, and really try to put those two data sets together. So what was nice was there was a lot of information publicly available about KMS11 through COSMIC, through CCLE, as well as our own data sets, so we could really compare and show what was happening with the BioNano Genomics technology. And so this just shows you some of the complexity that exists in these cell lines. You can see over here with RPMI 8226, there's tremendous complexity. Many of these are false positives. Many of these structural rearrangements are false positives. I don't think anybody would want to put their graduate student or postdoc through the ordeal of trying to characterize all of those. MM1S was to a lesser extent. And we chose KMS11 actually because it was very well characterized that it had two of the most commonly known translocations, the 414 and the 1416 translocation. And so it was an ideal cell line to test, and it also had a series of copy number abnormalities that were very well characterized. So we felt like it was the right cell line to choose. And so this is actually what the overall view of the KMS11 genome by BioNano looks like. You can see the resolution is actually pretty high. There are some gaps, but in general, there's very good coverage, 86% coverage of HG19. And this essentially shows you one of the first abnormalities that we had that BioNano went back and looked at. And so this is a very well characterized deletion in KMS11 on 11q. You can see here, this shows KMS11, MM1S, and RPMI. So this was sequencing. You can see there's this big gap and the lack of reads within this region. We went back and actually designed primers and confirmed that there was fusion between this end and this end. And this deletion fusion very well characterized. It was in Jonathan Keats's seminal paper in 2007 in Cancer Cell, showing that there was this interstitial deletion that created this fusion between these two genes. And so the BioNano Genomics technology was able to easily pick that up. They then went on and were able to show the translocation 414, which was very clear, as you can see by this map. So the translocation involves FGFR3 locus and IGH, and this is the region on chromosome 4P. This is the region on 14q, very well characterized in the literature. And it can be very difficult sometimes to piece together the reads with the IGH region within this specific translocation. And so we had the evidence by RNA-seq, and then we were also able to confirm it at the DNA level with BioNano Genomics. So this creates a very high level of confidence. It's proof of a concept, and then there was also the 1416 translocation that you can see here, another very well characterized translocation involving the IGH locus on 14 in this region on chromosome 16, and you can see the contigs are brought together very clearly. And then finally, there was this homozygous deletion on chromosome 17p, so there's a larger deletion of 17p, and the BioNano Genomics technology could resolve both the larger deletion that includes about 10 megabases, as well as this homozygous deletion of about 45 kb, shown here at the array level in KMS11. This is MM1S, which doesn't have the deletion, and you can very clearly see that there's a deletion here. And so for us, we feel like this was a great first step. We're now in the process of trying to run some additional samples so that we can confirm that what we're seeing at the RNA or DNA level by array or karyotype or next generation sequencing, the BioNano Genomics technology is also able to consistently find. With this KMS11 data set, it's been incredibly robust. It's just a matter of kind of where you ask. There's so many changes that are happening within the cell line that it's kind of an infinite number of questions to ask, and we're very excited about this and want to get this project completed soon so we can share the data with everybody. And with that, I'm very grateful for all the people at Emory that I work with, including Scott Newman, who's done a lot of the analysis, as well as Larry Boyes and Sagar Lonial and our clinical group, including Ajay Nuka and Jonathan Kaufman. And I want to thank everybody at BioNano, Gina, for helping us get this started. And I think this is a really important and big step in the process of trying to do personal genomes. I think a lot of us struggle with being able to pick and choose the right technologies and right combinations of technologies, and from what I've seen so far, I really think that BioNano Gina is going to help us confirm both structural rearrangements and copy number abnormalities and translocations in human genomes so that we can really advance the field and figure out what's going on in a number of different cancer types. And with that, I'll hand it back to Harper. And if there are any questions.
Okay. Thanks, Mike.
Sure.
Let's see, check the Q&A section. I think people have been pretty quiet during here, and you unfortunately just answered the question I was going to ask in your conclusion. So let's see what, can you pull up the Q&A panel? 1 second, go ahead and type in your questions, everybody. There should be a Q&A panel that you can access. Yeah, Mike, so I guess some people are answering in the Q&A that they don't have questions. So I have a question for you. I guess you showed a couple of examples of translocations. One was balanced and one was unbalanced. I wonder if you can kind of fill in a little bit more about the biological or the clinical implications between the two different types and I guess the value of being able to see those.
Yeah, so the translocations that happen with multiple myeloma and with many other cancers, it's really the fusion that's important. And so when you're sequencing cancer genomes, a lot of times you're not, especially if you're trying to do it to try to get at some clinical or biological picture, you're not so interested in what's happening in the background, so there will be lots of fusions and structural rearrangements that don't necessarily result in any phenotype. And so the ability to be able to detect exactly where two pieces of DNA meet is crucial. It's especially crucial if you know that you have a fusion and then like the 14-16 where you lose another segment, knowing kind of what the gene content is in those segments. And so I feel like what we're all struggling to do is find this right combination of technologies to be able to identify a lot of this, and these particular changes would require at least 2, if not 3 technologies. Many people are trying to get at this at the whole genome level. Usually there are depth of coverage issues, and it could get pretty tricky and pretty hairy, especially in dark regions. So I know that we see lots of translocations that happen, and it's not necessarily easy to map the other end just because there's high homology or redundancy. And so being able to know that there's 2 pieces of DNA meeting and what's lost or gained in between or upstream or downstream of that segment really helps us understand the disease.
Great, thanks, yeah. So we do have a question coming in, and the question is about, so I think it's somebody excited to hear a little bit more about the next steps, and the statement is that the validation shown here is great, and they're asking what are your thoughts about the discovery work that can be done with this, and maybe even kind of bridge that with talking about finding the 8X fusion that was shown with structural variation, and rather than validation, kind of showing that was a faulty call by the RNA-seq data maybe, and kind of tie that together with what your thoughts are for, I guess, moving this into a much more discovery realm.
Yeah, so for us, I feel like we want to continue to investigate the technology more, and the more data sets you run it with, the more robust you get to know how robust the technology is. So for me, I feel like there are lots of gaps in TCGA data sets, other public data sets, our own data sets. There are many false calls, false mapping, false alignments that happen in the more next generation sequencing you do with cancer genomes, the more you realize that. So there's this ability, this question of validation and what level of detail do you go through with validation. I don't know many people who have taken one complex cancer genome and then gone through and validated every single spot or every single potential change. It really becomes cost prohibitive and time prohibitive. You just do not have the ability to pull out everything, and so like many researchers, I think we cherry pick. The problem with cherry picking is that sometimes you pick the easiest stuff first, and that may not necessarily be the most biologically meaningful. So I feel like where we're going to progress with this is running a lot more BioNano experiments. And so we want to now try to run some patient samples that we've extracted CD138 cells from, that we're doing this comprehensive analysis by RNA-seq and copy number. We have the ability to go back and do whole genome if we have to, but I really feel like the biological meaning and being able to validate that there's some fusion present that produces a transcript, and then that it's easy to confirm that at the DNA level just moves the field much further along. And I think structural rearrangements are still, I think we've done a pretty good job with copy number abnormality, but structural rearrangements are still very poorly characterized in my opinion. I think there are lots of cancers that we really haven't fully investigated this with, and some of that is probably because of the limitations of short read sequencing.
Great. Okay, we have another question here. Have you looked to see if the cell lines you are working with are structurally stable? For example, compare the same lines obtained from different labs.
Yeah, so that's one of the beauties of it. So it's the reason why we didn't pick A226 because we know that line is horribly unstable. KMS11, as far as we can tell, the data has been consistent from the Keats data set to CCLE, and Cosmic data set. So there are at least 3 or 4 different laboratories, groups that have published genome scale copy number structural variation in KMS11. So at the mutation level, at the gene expression level, at the structural level, everything that we've looked at and found has been confirmed by other groups. It's one of the reasons why we chose that particular cell line to use this experiment with. So I know that several people that are in the myeloma community that have done a lot of genomics, it tends to be one of the best kind of test cell lines to experiment with, because they're well-characterized translocations and copy number abnormalities that we consistently see. So everything so far has been completely consistent with what's published and known.
Terrific. There's one that came in that maybe more appropriate for me to answer about the technology. The question is, what size ranges of structural variants are detectable with BioNano, and can rare events be detected with just a few cells of a sample? I'll give you a chance, Mike, if you have any input too. But in general, what we're seeing, and this requires, I think, a little bit more than simply the KMS line to really get at this question. What we know is the standard deviation of the positional accuracy of the labels that we're using to call these is 150 base pairs. And so with that, we're able to quite confidently get down into single-digit KB calls on deletions and insertions. We can probably go a little bit lower if you have a targeted CNV that you're looking for and are simply trying to confirm it, or kind of dive in and get a little bit more granularity. But on a genome-wide scale, we get a range from single-digit all the way up to multiple megabase size CNVs. In terms of rare events, and this would be in a case of a heterogeneous sample, this is an area where our algorithm teams are working diligently to improve the automatic calling of this. You would see these events in the single molecule data for sure, but that's sort of a manual approach right now to find the individual molecules that look different from the others. But we're working on that ability to do that automatically, and we'll have that in the near future. So moving on to another question that came in. Can you tell us what fold coverage you used? Mike, I'm not sure if you remember. I think it was 50 to 55, somewhere in there. Do you remember?
Yeah, I think that's about it. So is that a question at the RNA-seq level, or is it at your level, at the BioNano level?
It didn't clarify. Go ahead and answer the RNA-seq side. We'll have
the RNA-seq, we did, so at our end, we did about 50 million reads. At your end, you guys went back and ran additional sequencing. We probably, between the two of us, have gone well over 100 million reads. Yeah, it's hard to say at the RNA-seq level, because it really depends on the level of expression. But for us, everything that because these have been so well characterized, we spent most of our time looking at some of the low-lying fruit first, all the stuff that was really known, and then trying to go back and confirm or verify some stuff that we saw that might not have been as well characterized.
Perfect. Okay, so we have one here. So you were talking about combining technologies to get your answer. Does BioNano make very long reads as PacBio unnecessary to answer such questions about structural variations, or would you still find such data useful?
Yeah, so I'm the type of person that thinks that all data is useful. So I would not say that PacBio is not useful. If you're doing some model organisms or some infectious agents, PacBio can be incredibly useful. The thing is that a lot of times at the read level, you're not really interested in being able to see, everything that aligns perfectly. You just want to be able to detect that there's a copy number abnormality and exactly where the break happens. And for us, for what we do, I don't think that PacBio is very useful. But for some other applications, it could be. I could definitely see an instance where you might be sequencing an infectious agent and use a combination of PacBio and this, just because you always want that level of resolution and confirmation that there are things that are always missed at even with long-read technologies. And in human stuff, PacBio just has not been very useful for anything that we've done. But with other applications, it can be.
Great. So we're getting close to the end of the hour, but we have, I think, 3 more questions, 2 for you and 1 for us. Okay. There was a question about, is the genome assembly reference-based or de novo? And so we did the assembly here, so I'll answer that. The assembly is fully de novo. And so can you talk a little bit about the assembly process? So briefly, it's a custom-built algorithm for our specific data type, but based on the overlap layout consensus paradigm that has been used, for years in the Sanger days before de Bruijn graphs became popular. And that algorithm lives within our software, and you can just pull your data in and run this assembly and generate the answers. So with that, the last couple of questions. There was a question about false, the false positives that you mentioned, were they confirmed by BioNano maps?
Yeah, so that's a tough question. So the absence of evidence doesn't necessarily prove anything. So yeah, everything that we thought was a false positive, we didn't see any evidence for a rearrangement by NanoGeno. So far, everything that we've seen has been consistent. So if there was a fusion that we called that we felt was positive, we saw evidence by copy number array. BioNano actually resolved it better for us than what we saw at the copy number array level. Some of the stuff that we thought was false positive, we didn't see it by BioNano, and so that it's kind of a difficult question to answer. But I can say that we haven't had any major inconsistencies.
Good, thanks. And then I think on the last question, a little bit more open-ended and opinion-based. Someone's asking, what do you think is the eventual clinical utility for a technology like this?
That's huge, and that's so this is where I think anybody out there who does clinical stuff like myself, my goal in life has always been to replace the karyotype. I just have always felt that it was a very low resolution technology, and we never really understand genes at the G band level. I think the best place to start using this is really with hematological malignancies. I think the more places that can get access to these instruments and start running human hematological malignancies, AML, a number of leukemias, lymphomas, that all have structural rearrangements. I think this could be very useful. I think it could be run in a very similar time frame to FISH, hopefully much faster than karyotype. I know a lot of clinicians have not been able to fully embrace kind of what next generation sequencing technologies bring, because so much of what dictates clinical courses based on translocations or copy number abnormalities. Arrays haven't been fully adopted on the clinical side, so I really feel like this is a technology that can really kind of bridge that gap into the next generation of technologies. And for us, I think it's an ideal complement for things like RNA-seq and whole exome, where we're really trying to understand both mutation as well as structural abnormality to really kind of put the cancer genome in a complete context. And because there's so much dark matter in the genome, I think that there's going to be a lot that we can discover with this technology that we've just been missing, and you don't know until you actually start to generate that data.
Perfect. Thanks. So we've got to shut things down. There are a couple more questions that came in. We'll address those via email. Sorry that we didn't get to everybody's, but thank you, everybody, for attending. I did want to point out, I see there are several people that may be interested in plant genetics. I just wanted to point out we are having a workshop and a booth at Plant and Animal Genomes here in San Diego in January. So we'll have a host of guest speakers talking about a variety of different plant projects in case your interests span beyond cancer into things you can eat. So with that, please, anytime, send us questions. If you think of anything, we'll direct those to Mike as appropriate and get you an answer, whether it be about our technology or about Mike's research. We're happy to answer any questions. Thank you, everybody, for attending, and we'll talk to you again soon. Bye.
Bye.